Recombinant DNA, rDNA technology, is a sophisticated method that combines two different DNA molecules to study, modify, and use genes for various applications. This process, known as gene cloning, hinges on two critical components: a compatible host and a vector. Hosts in rDNA technology can be either prokaryotic, like bacteria, or eukaryotic, like yeast. These hosts are selected based on their ability to easily accept rDNA without destroying it, and to support the replication and expression of the inserted genes. The most commonly used prokaryotic host is the E. coli K12 strain, favored for its rapid reproduction rate. Other hosts include Bacillus subtilis, used when the goal is to secrete the protein encoded by the cloned gene. Whereas vectors are DNA molecules that carry the target gene into the host. For a DNA molecule to serve as an effective vector, it must be small enough to enter the host cell easily and contain a site of origin for replication, ensuring it can multiply within the host. It should also have unique restriction sites to prevent the vector from being cut into many pieces, as well as a selectable marker, like antibiotic resistance, to identify successful insertions. Before we move ahead, make sure to subscribe to our channel to never miss any updates. Today, we will be summarizing the chapter Host and Vector System from the book Biotechnology by NCERT, prescribed for Class 12th reading. This is Chapter 2. You can find Chapter 1 linked in the description below. This series contains all the chapters from that book. Moving on. The plasmids are small, circular pieces of DNA that are not part of a cell's chromosome, but can replicate independently within bacterial, archaeal, and some eukaryotic cells like yeast. These DNA loops range in size and can carry genes that benefit their host cells, such as those providing resistance to antibiotics. This makes them valuable tools in gene cloning, where they are used as vectors to carry and insert specific genes into host cells for study or to produce useful substances. They can be categorized based on their ability to integrate into host chromosomes, called episomes, or by the benefits they provide to the host, like drug resistance, R plasmids, or the ability to produce toxins to kill competitor bacteria, col plasmids. They also differ in replication behavior. Some replicate independently of the cell's chromosomal DNA, allowing for multiple copies within a cell, high or multi-copy plasmids, while others replicate synchronously with the chromosomal DNA, existing in only one or a few copies, low or single-copy plasmids. For gene cloning, plasmids are engineered to enhance their utility by adding features like selectable markers for identifying successful gene inserts and multiple cloning sites for inserting foreign DNA. One of the first and most famous plasmids modified for gene cloning was PBR 322, which was designed to be smaller and more efficient than its predecessors. Further advancements have introduced features like blue-white selection to simplify the identification of cells that have successfully incorporated foreign DNA, exemplified by the PUC19 vector. In short, we can say plasmids are versatile, replicable DNA fragments used in biotechnology as vectors to insert and propagate genes within host cells. Their natural properties, combined with genetic engineering, make them indispensable tools in the field of molecular biology for gene cloning and the production of recombinant DNA. Now, we shall discuss role of bacteriophage as vectors. Bacteriophages or phages are viruses that infect bacterial cells and serve as another key type of vector for gene cloning. Compared to plasmids, phage vectors are particularly useful for cloning large DNA segments and for screening vast numbers of samples more efficiently. Among the various bacteriophages, lambda and M13 phages are the most commonly utilized in creating vectors for E. coli hosts. Lambda phage vector. The lambda phage, which specifically infects E. coli bacteria, is a popular choice for a cloning vector. It features a head and tail structure, with the head housing the phage's genome. When the lambda phage infects an E. coli cell, it attaches to the cell surface and injects its genome into the bacterial cytoplasm through its tail. Lambda phage can follow two paths in its life cycle. Lytic cycle. In this process, the lambda DNA replicates within the host cell, leading to the production of new phage particles. Eventually, this results in the lysis, breaking down, of the bacterial cell and the release of new phage particles. Lysogenic cycle. Alternatively, the lambda DNA may integrate into the host's chromosome, becoming a prophage. In this state, the prophage coexists with the host, not causing any immediate harm and replicating alongside the host's DNA. This dual life cycle makes lambda phage a versatile tool in biotechnology for gene cloning, allowing scientists to propagate large DNA sequences and study their functions within a bacterial host. Make sure to subscribe to never miss any update on biotechnology. You can also tip us for our effort via this UPI QR. Let's fall back to our video. Now we move on to the bacteriophage M13, a filamentous virus that infects E. coli cells containing the F plasmid, has a single-stranded DNA genome about 6.4 kilobases long. This genome encodes everything needed for the phage's life cycle, except for a non-essential intergenic region. M13 attaches to and infects the host cell via the pilus. Inside the host, M13 DNA replicates to form a double-stranded intermediate, which then produces new single-stranded genomes. These are packaged into new phages that exit the cell without lysing it. The M13 vector, using this replicative form, allows for cloning of large DNA fragments and includes vectors like M13MP18, which offers a blue-white selection method for identifying recombinants. You should pause at regular intervals to make sure you get all the information. Digesting is more important than ingesting. We shall now talk about cosmid vectors. They are hybrid vectors combining plasmid and lambda phage characteristics, including lambda's SEOS sites for packaging into phage coats, allowing them to carry large DNA inserts up to 45 kb. These vectors replicate like plasmids within the host cell, but can be packaged into lambda phage coats in vitro for infection. Next we have phasmids or phagimids, representing another hybrid vector, combining aspects of both phages and plasmids. They contain lambda phage ends necessary for lytic infection and a middle region of linearized plasmid DNA, maintaining both lambda and plasmid replication functions. Phasmids can replicate as plasmids or phages within E. coli, depending on specific genetic conditions, such as the presence of a lambda repressor gene. In short, M13 phages, cosmids, and phasmids are all valuable tools in genetic engineering, each offering unique advantages for cloning and studying large DNA sequences within bacterial hosts. These vectors have revolutionized molecular biology by facilitating the manipulation, propagation, and analysis of genetic material. Next, we have eukaryotic host vector system. When it comes to analyzing the genomes of higher organisms, scientists face the challenge of cloning much larger DNA fragments, especially since eukaryotic genes often contain introns that can extend hundreds of kilobases. This complexity exceeds the capacity of typical plasmid vectors. To address this challenge, artificial chromosomes have been developed, capable of carrying extensive lengths of eukaryotic DNA. One of the primary eukaryotic hosts used in this context is the baker's yeast, Saccharomyces cerevisiae, which is advantageous for several reasons. Yeast cells can multiply both asexually and sexually, have a well-mapped genetic structure, and are considered safe for use, GRAS status, due to their long history in food production. Notably, yeast contains a 2 plasmid, forming the basis for various vectors designed for cloning large DNA fragments. Among these, yeast artificial chromosomes, YACs, stand out for their ability to incorporate DNA inserts ranging from 200 to 500 kb. YAC vectors are uniquely structured, including yeast telomeric and centromeric sequences, an autonomously replicating sequence, ARS, and selectable markers. They exist in both circular forms for bacterial growth and linear forms for yeast propagation. Besides YACs, other high-capacity vectors include bacterial artificial chromosomes, BACs, and phage artificial chromosomes, PACs, derived from bacterial plasmids and phage, respectively. These vectors vary in their capacity to carry DNA inserts, with plasmids handling up to 10 kb, bacteriophages up to 25 kb, cosmids up to 40 kb, PACs up to 300 kb, BACs up to 300 kb, and YACs accommodating the largest inserts of 200-500 kb. This array of vectors, each with different capacities, offers researchers a toolkit for cloning and analyzing genetic material across a wide range of sizes, essential for advancing our understanding of complex eukaryotic genomes and facilitating genetic engineering projects. Beyond these, we have expression vectors that are specialized cloning vectors designed not only to carry DNA inserts, but also to efficiently express the inserted genes. These vectors can be based on plasmids or viruses and are equipped with strong, often inducible promoters to control the expression of the cloned genes. Key features include unique restriction sites for gene insertion near the promoter and a proper transcription termination sequence near the 3' end of the gene, ensuring the accurate ending of transcription. The setup of a promoter upstream and a terminator downstream of the inserted gene forms an expression cassette, sometimes referred to as a sandwich expression vector. The success of gene expression using these vectors depends on both the gene's origin and the host used. For heterologous gene expression, where a gene from one type of organism, eukaryotic, is expressed in a different type of host, prokaryotic, the eukaryotic gene must not contain introns, as prokaryotes cannot splice them. Additionally, the expressed protein should not require post-translational modifications, like glycosylation, which are absent in prokaryotic hosts to retain its functionality. Shuttle vectors are another type of vector designed to replicate in two different host systems, allowing them to shuttle between prokaryotic and eukaryotic cells, including bacteria like E. coli and various eukaryotic organisms such as yeast, plants, and animals. These vectors have two origins of replication, but only use the one appropriate for the current host. Constructed through recombinant DNA techniques, shuttle vectors are versatile tools in genetic engineering, enabling the replication and expression of genes across different biological systems. Okay, that was quite a mouthful. Next video will deal in gene cloning. See you in the next video.